Welcome everybody to today's webinar. Our topic today is an introduction to OpenEye applications and toolkits. And our presenter is David Hamilton, my colleague from the OpenEye Application Science Group. If you got any questions during the webinar, please put them into the Q&A box on the top right of your screen. We will try to answer as many as we can during the Q&A session after the webinar. And if we can't get to it, we'll send you answers by email. Without further ado, David.
Thank you, Gunter. Hello out there, everyone. My name is Dave Hamilton, and I'm an application scientist here at OpenEye. Today's webinar is meant to be a high-level introduction to OpenEye's applications and toolkits that have found broad adoption across the drug discovery enterprise and beyond. As you will see, we offer a wide variety of software for cheminformatics and molecular modeling, and taking a deep dive into each application would occupy far more than the 20 minutes or so allotted to this webinar. So we will be keeping today's talk a bit cursory in order to give users new to OpenEye a broad look at what we offer, and experienced users of OpenEye's tools a reminder about all of the solutions that OpenEye has to offer. So let's begin. Before we begin, it's important to highlight who we are, what exactly our toolkits and applications are, and how they differ from one another. OpenEye has been in existence for over 25 years, and our mission to revolutionize computational drug discovery with physics-based, scientifically rigorous software aimed at advancing efforts to create new and better therapeutics, expanding as time has gone on. While today's talk will be largely driven by our applications, it's important to note that OpenEye got its start in this field over 25 years ago by creating programming toolkits for cheminformatics and molecular modeling. While we continue to offer our toolkits for licensing to this day, over the years we have also built user-friendly applications for use out of the box that leverage these packages of code. Named user licensing these applications and toolkits gets unlimited use of these applications and can run calculations either locally using your own resources or your on-prem resources, or you can run these applications in the cloud using your own cloud architecture. Recently, OpenEye has developed Orion, which is a cloud-native, fully integrated platform for molecular modeling and drug discovery, which enables users to seamlessly run our applications in the cloud, as well as to access and develop new science that takes advantage of Orion's ability to parallelize calculations. Despite the fact that we've been developing lots of new science that leverages this platform, it's important to note that OpenEye is investing more resources in and has more people working on our application and toolkit development than ever before. After all, our toolkits truly form the basis of the science that we offer. Our downloadable applications are targeted towards problems related to early-stage drug discovery and can be used both during hit finding and optimization phases of these campaigns. Over the course of this talk, I'll be sure to highlight applications that can be used for hit finding in different contexts, leveraging information both about known ligands and structures in different ways in order to find diverse ligands that form the basis of discovery campaigns. We'll also talk about our tools that can be used for hit-to-lead or lead optimization as teams seek to optimize binders into potential candidates for the clinic. Ultimately, our goal with our modeling tools is to find active compounds, and for 25 years, the focus of our toolkits and our applications have been to accurately predict what we feel are the two primary descriptors of a molecule, shape and electrostatics. Ultimately, these properties are what we feel dictates the complementarity of a particular molecule to an active site of interest. Our code, as such, is written in an effort to be able to do this accurately and, almost as importantly, quickly, in an effort to allow our users the ability to model as many compounds as possible as quickly as possible. And as such, our code base has long been recognized for its reliability in providing users with actionable insights as quickly as possible. And as such, licenses to our software do not restrict you on speed or scale of calculations. In my mind, it is easiest to understand the breadth of our solutions and the many problems that they can solve by looking at our software through the lens of the applications that we offer, which in and of themselves cover a wide range of areas related to molecular modeling and computational chemistry. As such, I've broken this harbor tour of our applications up into 3 different use cases that will be of interest to our audience. First, we will discuss the applications that we have available for cheminformatics, in which we are interested in the manner by which we read and write molecules, as well as handle their molecular graphs. Second, we'll take a look at the applications that we have available to leverage 3D similarity to help users explore chemical space to find actives on the basis of shape and electrostatic features. And finally, we'll look at our tools for biomodeling and structure-based screening and design, which also depend heavily on our ability to accurately model shape and electrostatics. Before we get started with our applications for modeling downstream, we'll get started with our cheminformatics offerings because, and I'll use a favorite quote from Anthony Nichols, you can't do good modeling without cheminformatics. This just makes sense. Before we can model compounds, we need to know what to model, and as such, we need applications to help us appropriately read and write molecular databases in a variety of formats and have the ability to look at and manipulate chemical graphs and triage ideas quickly. As such, I'm going to get started by highlighting not an application, but rather a toolkit. OEChem has long been recognized for its accuracy, flexibility, and speed when it comes to reading, writing, and manipulating databases of molecules found in a variety of file formats, including popular formats such as SDF, SMILES, Mol2, INCHI, PDB, and CIF, as well as OpenEye's proprietary formats OEB, OEZ, and OEDU, which contains design units that contain target structures. OEChem, along with our other toolkits, powers our applications in the background, but if licensed, allows our users to write powerful scripts for many flavors of cheminformatics tasks in molecular handling. The first application that I would like to touch on from the world of cheminformatics is an application called Filter, which does exactly what its name would suggest. It filters small or large lists of molecules quickly on the basis of filtering rules provided from a simple .txt file. This file can contain rules for filtering on the basis of a variety of computed properties, including such criteria as molecular weight, heavy atom count, number of rotors, PSA, X-log P, and so on and so forth. Importantly, this functionality also extends to substructures and functional groups, many of which are hard-coded into moieties in the toolkits that can easily be called in the filter file, while still affording the flexibility to encode your own moieties with smarts patterns. Alongside the application, OpenEye offers a number of filters out of the box, including blockbuster, lead, drug, and panes. And these filters can also be modified easily for use across projects. Like many of our applications, filter is called from the command line for easy and expedient use. As a companion to these capabilities, a simple GUI application called Picto allows users to perform a number of cheminformatics tasks in a visual environment. While Picto was originally written to be a simple sketching tool with translation capabilities to visualize SMILES or generate names of molecules on the fly, we've recently given the application an overhaul that allows users to test smarts patterns and visualize matches on the fly, or to manually generate atom mappings for use in developing smirks for molecular transforms. We envision Picto as a useful Swiss Army knife for cheminformatics development. And as a final highlight of our cheminformatics offerings, QuackPack is not one application, but actually several smaller applications that can each be called from the command line. Tautomers is an application that is capable of taking an input database of molecules and enumerating tautomer states of those molecules for modeling downstream. FixPKa likewise does what its name would suggest and allows users to either apply a neutral pH model to a database of molecules for modeling use downstream on the basis of smarts-based heuristics, or to remove ionization states of molecules when possible to change molecular representation to a format that is more compatible with the way that chemists typically visualize molecules. And finally, the application MolCharge has the ability to take inputs in 1D, 2D, and 3D and assign partial charges to the atoms within the molecule. This application supports AM1BCC, MMFF, Amber, and Gastiger charge models. Of course, we're not just interested in cheminformatics here at OpenEye, we are also interested in all of the important modeling downstream that is finding increasing impact in the world of drug discovery programs. We'll start our tour of our modeling applications with the applications relevant to problems related to 3D ligand similarity. That is, we are interested in predicting databases of reasonable 3D shapes of molecules, also known as conformers, in order to search through them in order to predict a particular compound's behavior with respect to a target of interest, and find new active chemical matter on the basis of 3D similarity. Before we can mine for 3D similars to a molecule of interest, first we need to be able to generate such conformers of interest. For these purposes, we created a program called Omega. Omega utilizes curated fragment and torsion libraries in order to quickly generate large ensembles of reasonable conformers for compounds within a given input file, and then prunes those ensembles down on the basis of deduplication criteria and force field evaluation of energies, along with other user-provided criteria. As such, Omega is an exceedingly fast and flexible conformer generator while maintaining a high degree of accuracy, making it a top choice for the creation of multi-conformer databases of molecules for downstream modeling. One such use of Omega downstream is a program called ROCS, which stands for Rapid Overlay of Chemical Structures. ROCS generates what is known as a query from a provided structure with 3D coordinates, which can be harvested from a public or proprietary crystal or cryo-EM structure, from a pose generated with docking or posing, or even just a single conformer of a molecule generated with Omega. This query encodes the shape of the molecule, along with the chemical features of a molecule that could be relevant to binding, such as hydrogen bond acceptors and donors, anions, cations, rings, and hydrophobes, as color features. Both shape and color are encoded as three-dimensional Gaussians in order to enable rapid searching for similars throughout a provided database. Each conformer of each molecule within the database is then overlaid on the query and optimized for alignment with the query in order to attempt to maximize the overlap of both shape and color features between the two entities. Scoring is typically done using Tanimoto combo scoring, which evaluates the degree of shape overlap and color overlap from the database separately, and then these two ratios are combined to provide a final score in the range of 0 to 2 that represents the 3D similarity between the two entities. In the end, ROCS provides a rank-ordered list of compounds in order of their Tanimoto combo scores, along with the alignment of molecules from the database with the query. An example of these overlays is seen in the viewer on the left side of this slide, where we are scrolling through a series of molecules against a particular query from low similarity to high. Importantly, ROCS is very fast, which enables tasks such as ligand-based virtual screening without any context about a particular target. However, it is also important to note that ROCS is flexible, and parameters such as alignment method, scoring method, changing weights assigned to particular elements of a query, and even the color force field, which defines the color features, can all be modified by the user in order to carry out the search and alignment in whatever way they feel fit. Along these lines, EON is a program that is often deployed as a follow-up to ROCS in order to rescore the ROCS alignments with electrostatic fields calculated using the ZAP toolkit in place of the color heuristics that we use as a part of ROCS. This electrostatic Tanimoto can be used to help identify particularly unique compounds within a database independently or in conjunction with the other ROCS scores to further evaluate your hit lists and aid in compound selection. And finally, Brood is an application that is designed to leverage the strength of OpenEye's 3D similarity capabilities in order to make an impact on optimization campaigns. Brood allows users to select just a portion of a molecule for replacement in order to retain desired pieces of a molecule while replacing fragments for the purposes of scaffold hopping, R-group replacement, property optimization, patent busting, and more. Brood enables this by performing a 3D isosphere search on only the selected fragment by using shape and color, or shape and electrostatics, against a multi-conformer database of fragments that is provided by OpenEye. Ideas can then be filtered on the basis of predicted properties, strain, or potentially even clashes with a provided protein structure. Finally, Brood clusters the resulting ideas, allowing for easy exploration of the generated idea space. Users are also not locked into the provided database of fragments, as a custom Brood database can be created by using Brood's companion application, Chomp, and a set of structures from which fragments can be created. Finally, in the last section of this talk, I want to talk about OpenEye's solutions for biomodeling, structure-based virtual screening, and structure-based design, which holds an equally important position in OpenEye's package of applications, as are applications for ligand-based design, and these applications can also afford users access to diverse actives that may not be found by using ligand-based methods alone. Any discussion of structure-based design has to start, necessarily, with a discussion of the preparation of the target models that will be used in the overarching workflow. OpenEye's application for processing protein structures is an application called Spruce, which takes a target protein structure and outputs a modeling-ready structure known as a design unit, which can be used for docking, molecular dynamics simulations, and other structure-based screening and design methods. Spruce accomplishes this task by fetching data, flagging issues with the structure, splitting the system into components, including the protein, cognate ligand, packing residues, solvent, and more. The application then builds loops, tails, caps the structure, and appropriately places hydrogens around the target structure. Finally, the structure is evaluated for trustworthiness on the basis of an iridium score if the accompanying density maps are provided to the application. The end product is a model of a given target that is appropriately prepared for downstream tasks. The next application I'd like to touch on is an application called SiteHopper. This application was developed for polypharmacology purposes, such as finding potential off-target effects, target fishing, or drug repurposing. SiteHopper can be used to compare binding sites of many different protein structures. It accomplishes this goal by generating a patch of an active site of interest by creating a shape query out of the inner surface of the pocket and the color features of the various chemical groups that line the active sites. Much like in rocks, the query patch is then overlaid against the other patches in a database in order to find other active sites within the database that have high similarity and are likely to bind similar classes of compounds. Of course, central to our biomodeling and structure-based design solutions is our docking algorithms. All of our docking algorithms are packaged into a single application that we call OE docking, which enables virtual screening and beyond. For virtual screening, our docking engine is known as FRED, which stands for Fast, Rigid, Exhaustive Docking, which exhaustively samples the active site of a protein by taking every conformer of an input molecule, populating the active site with multiple copies of that conformer in various translational and rotational states, throwing away clashes with the protein, scoring with a light scoring function, optimizing the remaining poses, and then finally scoring with the ChemGauss4 scoring function. Alternatively, FRED can be run in hybrid mode, which is a docking process that is guided by cognate ligand poses. This docking algorithm is the same as FRED, except the first pass scoring function relies more on similarity to a cognate ligand pose than to the actual docking pose itself, and this helps to guide ligand docking towards a particular region of the active site. These methods are meant to be used for virtual screening, which the goal is to enrich an overall list of molecules with actives rising towards the top of the list and molecules likely to be inactive towards the bottom of the list. To aid you in this process, we have a utility that is called MakeReceptor that ships alongside OE docking. And MakeReceptor allows you to input your protein structure in OEDU format and customize the box in which an active site is located, the outer contour of the docking receptor that is generated, as well as to allow you to set constraints on your docking run in order to selectively keep only ideas that form the particular poses that you're looking to form. And finally, Posit is an algorithm that is used for generating reliable poses of molecules within an active site with a co-crystallized ligand. Posit first determines the Tanimoto combo of a ligand to be posed to the cognate ligand, and on the basis of this measure, selects a method for posing. For molecules with a high Tanimoto combo, an algorithm called ShapeFit is used, which flexibly fits the ligand into the cognate ligand in order to generate a high-quality pose. For poses with lower Tanimotos to the cognate ligand, and for which flexible fitting is not reliable, the Posit algorithm instead falls back on hybrid or FRED and docks the molecule instead. Another application that has found great use in structure-based drug design is an application that's called SHMAP, and its companion application, GamePlan. The purpose of these applications is to probe the electrostatics of an active site by using water. And by this, what I mean is we model the active site with a high dielectric continuum, and then we test the electrostatics of this active site by using probe water molecules in multiple orientations, both in their charged state and in their uncharged state. Now, SHMAP enables the exhaustive sampling of this active site. It also enables you to direct the various locations of the active site that you'd like to sample with these probe water molecules. But the companion application GamePlan actually samples testable hypotheses around the active site on the scaffold of the cognate ligand that is found within the structure, at locations 1 or 2 bonds away from the scaffold, or at sites with waters that you might want to consider displacing or conserving. And by using the information that falls out of both SHMAP and GamePlan, it enables you to develop some testable hypotheses for modifications that you can make to your lead scaffold. And in some published instances, has led to multiple fold improvement in the activity of the molecules that are being built and designed. And finally, I want to talk about the application SHIBKey and its companion application Freeform. SHIBKey is really all about molecular energetics using force fields, and SHIBKey can be used for such tasks as minimizing small molecules into an active site, and its companion application Freeform can be used for estimating the delta G of binding and the ligand strength associated with taking a small molecule in solution and binding it to a protein target of interest. And finally, a word on visualization, because all of these applications that we've talked about today, and many times you want to actually be able to evaluate the visual results of these calculations. And for these purposes, OpenEye offers a multi-purpose visualizer called Vita. It's a flexible viewer to support all products OpenEye and beyond. And Vita features high-quality graphics, preset views for viewing calculations from OE applications, a chemically aware spreadsheet for display of 1D, 2D, and 3D data, scriptable control of the interface, easy export of data, and more. And of course, I've tried to weave in the themes of our toolkits throughout today's applications-driven overview of our products, but it's important to note that all of the code that underlies our applications ultimately arises from our toolkits. And all of our toolkits for molecular modeling and for cheminformatics are available for licensing, and they come in C++, Python, C#, and Java. So I wanted to take a minute just to thank you for your time and attention and for coming to this webinar today about our applications and our toolkits and the various downloadable offerings that we have for you to purchase and license. Again, I've appreciated the opportunity to tell you all about this, and I'm looking forward to interacting with you all more in the Q&A in just a minute. Thank you very much again.
Hello, everybody. Thanks, David, for the presentation. We're now in the live Q&A. If you have any questions that you didn't post before, you should see on your platform in the top right-hand side a Q&A section to post more questions. So please feel free to send us more questions and we'll try to get to them either here or we follow up by email later on. I do have questions here, David. First one. Can filter handle custom-defined functional groups?
Yeah, you can define all kinds of custom filter rules and selection rules with smarts patterns directly within the filter file, which with the applications we provide those files so you can edit them as you feel fit. There's a wide variety of moieties that are hard-coded in the toolkits already, so you don't always need to come up with your own smarts patterns. And filter can obviously call those within the filter file, and these moieties are extensively and clearly documented on our website. And just in case you're not aware, if you're interested in perusing our documentation, we make that publicly available. You don't already need to be a user of our applications to actually access those. Those are publicly available at docs.eyesopen.com.
Yes. And some of these filters that we have in there are pretty, some of them look pretty exotic, standard, but then some unusual ones, I think, are predefined. So yeah, users don't have to do it all themselves. Next question is here. How does fix pKa assign pKa states?
Yeah, so we built a model for the assignment of pKa states on the basis of a smarts tree, and fixed pKa is heuristic based. It's not actually modeling exact or predicted pKa's for particular molecules. It's just relying on this smarts tree underneath the hood to make it really fast. But there is an application that I didn't mention over the course of the talk called pKa Prospector that also ships with the application package. And that allows the user to actually query our pKa database on which the model is based. And it's distributed with the package, as I mentioned before. So.
But that then is kind of a lookup for pKa's for groups
Exactly.
that you're not sure about. It's
yeah,
really a lookup for experiments, right?
Yeah, for substructures or for similar molecules and so on and so forth.
Can you, quote unquote, fix a portion of a molecule when using omega to generate conformers?
Yeah, you can do that. You can do that providing a fix file to omega to specify a substructure to fix and align to. And then there are a bunch of different flags that you can set within the application to loosen the RMS requirements, to fix on the basis of smarts pattern matching or on the basis of maximum common substructure. So you've got some control over how that fix process happens as well.
do I have to use spruce? You talked about spruce, right, to prepare protein. Do I have to use spruce to generate a protein for use in OE applications?
Yeah, so sort of, in the sense that our applications use design units that are created by spruce. So you do need to use spruce in order to actually make those proteins interface with our applications. But that being said, if you want to actually prepare your protein and control how that preparation happens with other software, you can. You just have to export the file as a PDB. But to render that PDB file compatible with our tools, you need to pass the PDB file through Spruce to create the design unit. But there's a flag that you can set. That flag is called no preparation, and that turns off any additional preparation that Spruce would do to that file. So the answer to your question is sort of. You can do that preparation elsewhere, but you do still need to actually pass it through Spruce to create the design unit.
Oh, another one here. What force fields do you support with ShipKey?
Yeah, so we support OpenFF force fields. We have both Parsley and Sage. For Amber, we have Amber FF14SB, Amber 99, and we also support MMFF 94 using ShipKey.
And I guess for the open force fields, whenever there's a new one actually officially released, we will right away support it as quickly with the next release. It will go into be supported by ShipKey, right?
Yep.
There is one more here. Can I change the docking scoring function?
Yeah, sure. So more flexibility in our docking scoring function is something that we've certainly considered. But at this time, that's not something that we support with our OE docking suite.
Okay, let's take a look at a few more that came in. I would like to evaluate Omega GPU. Who should I contact? Could you share your email info? I guess the general info is just either support@iceopen.com or I think sales@iceopen.com to check out Omega and get in touch with us. Usage depends a little bit on where you are and the licensing, but that's all handled then in the end by the support. They can forward you to the right place. So support@iceopen.com, maybe reference the webinar, and you can get information on using any of our tools. I need a detailed procedure to perform a docking study. There's a question about detailed procedure to perform a docking study from A to Z. I think that's something in the tutorials, in the documentation again, right?
Mm-hmm. That's correct.
So again, David, you mentioned before the docs.iceopen.com. If you go there and the docking part, there should be tutorials. And I think we tried to guide you through one example, at least from receptor preparation to the docking steps that are needed.
Yeah, and it's certainly something too that, if you contact support@iceopen.com with your specific use case, we can certainly get you some more information to help guide you in the specific protocols and workflows that you're looking to establish. So.
There's another question, maybe the last one, or we'll see. Sure. When using Omega, is there an easy way to generate tautomers/dominant protonation states during the conformer generation step such that it can be called with one command? I think the one command is on the command line, the maybe tricky part. It would be two commands. One calls tautomers. Mm-hmm. And the other one calls Omega on that file that was generated with the correct tautomers. But it's easily scriptable. So if you put that in a shell script, run the first one on the output, run the second command. It looks like one command to you. You just have to write a little script to automate the combination of the two. Where this is easier packaged might be in the Orion platform, if you actually were to go there, because there we have this, we didn't talk much about that one. There's a whole web platform that you can get from us. And there we have a kind of a pipelining tool where they can string things together. But that was a different presentation, or will be a different webinar talking about Orion more. That's where it might be more streamlined. But otherwise, the command line versions are very fast, very efficient, and and really very stable for us, working on it for 25 years. I think that would be the way to go.
Yeah.
There was one more before. We don't get to all of them, sorry. There was one more before by Stephen. We have to follow up with you because you asked for extracting the ligand out of a design unit. That will require a bit more of an email answer, Stephen, but thank you for the question. We'll certainly get back to you.
Okay.
With that, I think for today, thank you very much, David, for the presentation and the effort to put all this together. It's a lot of work. Thanks, everyone, for joining us, and we hope to see you next time.
Yeah, thank you all for coming.
Thanks, everyone.
Bye.